So indeed, I wanted to share an insight that we had at our lab. So we were using the VSHD, and when we got it, we were, I think, first users or one of the first users of the system, the upgraded new high definition one. And it took us some time because it appeared that it was more complex than the previous system that was available. So it took us some time to play around with it. And we wanted the stimuli to be more engaging, and that was basically what we were interested in. But of course, the VSHD comes with 2 cameras, and that gave us good opportunity to set up the goggles faster and better. With the previous version of the goggles, it was kind of hard. You had to either allow the person to adjust it themselves, but they are not always the best at that, or you had to just ask, do you see well? Here we could finally see both eyes, so it was very easy to objectively set them such that you know as a researcher that they are set correctly. There's no more guessing. It's faster and more efficient. So that was kind of the goal. But then I started a study that was about reward processing, about emotional processing, and well, pupillometry is it is a measure of what's going on in terms of arousal. So that when the pupil dilates or contracts, it can be related to emotional processing. So I started looking into it, and it's it turned out you can basically almost for free get a very interesting measure of what's going on with the person. And that kind of changed the way we started to design our experiments, just to include, for example, the luminosity of the stimuli and other aspects of them. So we didn't change much from what you would usually use, but that allowed us then to potentially compare in terms of the arousal of the participant. So it's gonna be maybe easier like that. So the pupil reaction is most of you probably first think you think is when the light is bright, your pupil contracts. When the light is dimmer, then it will become bigger. But what's important is that there are 2 systems in your body, sympathetic and parasympathetic, which are related to activity of 2 groups of muscles. And one of the muscle constricts it, and the other muscle dilates it. And it turns out that these 2 parts of the system are controlled mostly by different neurotransmitters and different parts of the brain to some degree, although the interplay is actually very complex. But in general, it's about interplay of 2 systems, that one is making you more aroused, one is making you more relaxed. It's like fight and flight versus digest and chill. So the brain area that is thought to be the first mover to some extent is this one, which creates most of the brain's norepinephrine, which then is a precursor to noradrenaline, and that then transports this neurotransmitter as well as just the activations forward to the subcortical and their cortical areas. And we know that this is an important factor in how the general arousal of the brain and therefore the processes that are going on happens. So as I said, it's the pupil response would be reacting to the brightness of the stimuli, but interestingly also the color of the light would also influence how much it constructs or dilates. Here the intensity of the stimulus was the same, but the color was different, and we see that the reaction is different. It will also that the pupil will constrict or dilate when you're changing your focus, which could be most easily imagined as looking through a window. If it's a rainy day, you can look at the buildings, but you can also shift your focus without too much thinking, for example, for the droplets that are just dropping. That will also change the diameter of the pupil. But I think the maybe most interesting for research and perhaps beyond would be the increase or the change in the pupil diameter in response to arousal, which could be mental effort, but it could be other aspects of arousal, and that would be the psychosensory pupil response. So even if there's no specific stimuli, there is a fluctuation in the pupil diameter, and that is called hippus. So when the pupils are small, that's the low arousal, and usually what that means is that the behavior is guided by the environment. So if something pops, some aspects of the sight you're seeing are, let's say, bright lights, sharp corners. But then when the pupils are large, that means that at least generally, that the arousal is higher, and therefore you would be more guided by the instruction of the task, if we're talking about specifically about tasks. So for example, if you're looking for keys, then that will be the something that will pull your attention towards it. And we know that the hippus fluctuation, that tracks physiologically the level in arousal, and we know that this can affect the dynamic of how we react to the environment in a subtle but measurable ways. And pupil dilation has been linked to activity mostly in Salen's network, which the main hubs would be the dorsal anterior cingulate cortex as well as bilateral insula. And this is thought to be increasing the performance in tasks. So now I would like to show you 3 examples of how pupillometry can be used in the research. So one would be in the reward processing field of research. Here the task was such that there was the, let me just try to, yes. So you saw a cue that didn't mean really anything, and afterwards you would be receiving a reward if you responded correctly. And some cues were tied to monetary rewards, some were to social reward, or the control ones. And the interest was to see how the pupil changes not during the outcome, so the actual reward that you receive, but rather when the cue appears and you already know what the cue will mean. And we can see that this is the response. So if there's no reward, then the pupil didn't really change too much, and monetary rewards, maybe not surprisingly, was the most arousing in this example. And this is which brain areas activity can be explained by pupil dilation, and that we see indeed that the Salen's network has been mostly activated here, as well as some deactivation default mode network. It can also be used for fear processing. So here was a different type of task. So you also saw some cues that didn't at first mean anything. So that was a conditioning task. So you had to learn what means what as the task progressed. And then you would just zap the person in a painful way. So therefore, people quite quickly learn which cue means that you will be shocked. So you had no shock, which is CS minus. Then you had shock with 60% chance and shock with 100% chance. So in general, we know that we see that the responses of the pupil, even for both types of chances for shock, were significantly higher. So that was the conditioning, but then researchers also did the extinction, which is the shock will no longer come. You will see cues, but the shock will not come. And then people learned that, at least to some degree, that the arousal was decreased, and that can be seen on this graph. So in the extinction phase, the pupil dilation significantly and pretty robustly, if you look at the standard deviation there, changed. And researchers also did the, let's say, correlation analysis, and you can see as well that indeed Salen's network was activated, but interestingly also in case of more arousing, perhaps stimuli, the parts more related to low-level processing of arousal were also seen here. It can be also used in cognitive effort studies. So here the task was to remember, so you were shown a stream of stimuli, let's say letters, and you have to remember which letter, what kind of letter was presented before the next one. So, and if it's the same, you have to react. If it's not, then you don't. So that would be one back. But then you can have a stimuli, let's say T, A, T. So now you have to remember that two stimuli back was also a T, and you would have to react. So therefore, you need your working memory to be more jogged for that. And we can see that indeed the working memory load can be explained with the pupil dilation as well. And here's how over time that fluctuated. So those were blocks, 40 seconds blocks, in which there was just barrage of stimuli. Similar analysis and very similar result on the brain level. So this is very robust, I would say, between not just studies but also the tasks and cognitive domains that they were studying. So in 2023 on OHBM there was a whole day educational course which was about physiological signals. How to account for it, what does it mean, should we get rid of that, should we model that, should that be the part of your understanding of what's going on. And similarly in last year's OHBM it was very similar, half a day this time. Interestingly, 90% of talks and topics were not about pupillometry, were about other physiological readouts. And I asked in 2023, then I asked in 2024, what about pupillometry? We know it's a very unique and accessible to some degree, at least metric. And the answer that they got me is that this is something we have to look into, but it's just less popular. Nevertheless, I feel like one of the reasons is actually people not having the capacity to do it, because there's just no equipment. And usually if eye tracker is used, it would be used for specific eye tracking questions. So there's just few studies using actual pupillometry in fMRI. Here I wanted to share with you some of our projects for which we did collect the data for pupillometry, maybe as an example of how that can be used. So in this project, which this is the thing I'm going to defend next month, so that was similarly conditioning task in people who were quote-unquote addicted from pornography. And here is also you have a cue that means nothing at first, and you learn what kind of reward comes afterwards. And then you learn as the time progresses. And here what I have is the reaction time. So I have a behavioral measure. Subjects were reporting, self-reporting how arousing were the stimuli, but not the outcomes, but rather the cues. So before they enter MR, I ask, how do you feel about circle? It's not arousing, right? About triangle, not arousing. But after the conditioning, you can see here, this part after the conditioning, all subjects, although the pornography addicts had a positive valence and arousal towards the cues themselves. I, of course, have the neuronal reactivity, but then I can have the pupillometry and therefore the arousal, and therefore seeing the evolution of how the learning in terms of arousal that it puts these subjects in evolves. Another study, my colleague Agnieszka already told you some about it, so I'm not going to repeat, but what I wanted to say that this is also a example of study that we want to have as many levels of description of what's going on in these problems as we could. So we had data on everyday life. These patients have for 7 days wore a watch that was gathering the data, and they were also asked from time to time to answer a few easy questions. We had the self-assessment, which would be this part of the task, so how aroused were you by the stimuli. We had the behavioral, so this is the task that was supposed to make the emotional reaction go away because you're just focused on math. So we can see how, if you were aroused emotionally, in a negative especially, you would be less cognitive processes will be available to you. So the reaction time should be longer and more mistakes. If you're upset about something, you might not solve the equation just as well. And then the physiological, which would be or psychophysiological, which would be the reaction of the pupil during this time. And then, of course, the neuronal level. Another study, this was on healthy, at least there was no indication otherwise, seniors, young seniors, so 50 to 60 years old. But they were screened in terms of the genetics, and they were invited based on the genetic factors that predisposes for Alzheimer's disease. So we are curious, can we find some sort of biomarkers or correlates before any onset? So they filled in many questionnaires that were about finding whether there are any cognitive decline, and they were basically fine. And for that, we also gathered the data. First, we didn't do that, but I found out later that it might have been interesting to do so. Is that we could have got just the response to light as a proxy to the acetylcholine system, which we know is mostly reactive towards the response of the light itself on the brain. But then also, of course, as they were, the subjects were doing several cognitive tasks, as I said before, for example, the working memory load, but generally how much effort they have to put into solving anything, which these tasks are quite challenging, I would say, can also be a measure of that, just how hard those tasks were for these. And this is not yet a reality. This is something we applied for, so we were waiting for a decision. And here we wanted to see what is the emotional regulation mechanism in behavioral addictions. And for that, this is an interesting task in which you stress people. You put them into social stress situation. And here also we want to see what is their cortisol level before getting into MR, after being stressed, and afterwards as a follow-up. And of course, other measures that I spoke about before to have as full picture of what is going on as possible. In many papers, we can see, for example, just brain results, just reaction time, and usually doesn't tell the whole picture. But it's also a reason for that, which I have to share. I don't like that about where the science is going, to be as concise in the paper as possible. If something is not necessary for the story you want to tell, you should throw it out. I don't think that. I think that if you can have more comprehensive picture with, for example, many readouts, I think it makes more compelling evidence for what you're studying. Thank you.